All right, next we're going to do active and passive transport. So what we're asking is, are we able to move something across the cell without using energy? That's really what we're asking here. If the cell doesn't have to put in energy, and this is a double layer, okay, so this is like this is a double layer plasma membrane, but I'm just going to do one from now on just so that we can get this. We don't want to clutter our pictures. So this is outside the cell, this is inside the cell. I have some integral proteins, maybe a peripheral protein, some glycolipids, and some cholesterol, and that kind of stuff. So we know we're at our plasma membrane. If I want to move something outside the cell to inside the cell with no energy, so no energy, this is called a passive transport process. Does not require any energy investment from the cell at all, okay? To be able to do this, you typically move from an area of high concentration to low concentration. So high to low, like that. When you move from a high to low, or you move down a concentration gradient, or with a concentration gradient, all these terms mean the same thing. So we can say high concentration to low concentration. We can say down a concentration gradient, and this is concentration right there. These brackets are concentration. So down a concentration gradient, or we can use the word with a concentration gradient. So that's passive. These are all passive transport. What I'm getting at with all these stuff, this is what you should look for on a test. On a test, you should look for high to low. You should look down or with the gradient, and you should look for no energy. All those are cueing you into these passive transport processes. So now let's look at the different forms of passive transport. Let me write some stuff up, and then I'll restart the video. All right, so these different types of passive transports. We have diffusion, we've got facilitated diffusion, we've got osmosis, and we've got filtration. Let's start with diffusion, and this is also called simple diffusion. So if this is a passive transport process, what kind of concentration gradient are we moving from? We're moving from a high to a low concentration gradient. I was giving you some time to quiz yourself there. So we have a high to low concentration gradient. What else do we want to know? What affects simple diffusion? So things like size. We talked about this before. Size matters. What's going to move through the plasma membrane more easily? The small 20-pound kid or the 6'4", 240 linebacker? The smaller kid's going to move through that tube quicker. So just like that, these smaller molecules are going to move across the plasma membrane quicker. So larger things take longer to diffuse across smaller things. Temperature, go ahead and ask yourself this one. You think increasing or decreasing temperature will increase the way things move across the membrane? It's going to increase, because when we increase temperature, what we're really doing is increasing the velocity of the particles, so they're moving faster through that membrane. Next is polarity. So we've already talked about this before. Which one of these substances, polar or nonpolar substances, would be able to simply diffuse across a plasma membrane? So nonpolar substances can diffuse freely across a plasma membrane from an area of high concentration to low concentration. So no energy is required, but the molecule has to be nonpolar. Okay, facilitated diffusion, again, doesn't need energy, but it's typically because the molecules are too big to just go straight through the plasma membrane, so they need some help. So if we have our PMEM, our plasma membrane, and we have this integral protein, and it has a channel through it. So something, we're just going to call it compound A for now. So compound A, it's nonpolar, but it's really, really big, so it can't do simple diffusion. But it's nonpolar, so it's not needing energy to get across this thing. It still wants to go from an area where there's a ton of A's to not that many A's. So still high to low concentration. But we need this carrier protein. So when you use a carrier protein without using energy to move something from high to low concentration, that is facilitated diffusion. What are some things that affect the rate of this guy? So if I have my integral protein, what's going to affect things going through there? Well, there's a few things. The concentration gradient. So if we have a really high to low, that's going to increase movement diffusion across this carrier protein. Okay, if we have small high to low, then we have small movement across the concentration gradient. Small movement across the membrane if you have small concentration gradient difference. Okay, so if we were doing that A thing and we were on to ask ourselves, based on the amounts, based on these amounts, which side is going to, and this is side 1, and this is side 2 over here, which is going to have more instances of facilitated diffusion? The thing that has more concentration gradient difference or less? We just said more, okay? So there's a bigger difference over here compared to over here, and we want to reach this equilibrium where we reach like our homeostatic range. So this will come through at a faster rate for this side. Okay. So facilitated diffusion, again, the concentration gradients. Things move from a high concentration to a low. So if you increase the difference of this gradient, you get more diffusion. The other thing is, well, how many integral proteins do you have? Do you have 1? Do you have 2? Do you have 3? As you increase the number of channel proteins, PTN is going to be protein right now, then you increase facilitated diffusion, and that makes sense. If everything has to wait its turn to go through, well, if you have 3 different things something can go through versus just the 1, then facilitated diffusion is going to occur at a faster rate. So the 2 things, concentration gradients and amount of channel proteins. The last one is the pickup speed of the carrier protein. So when I'm here, this carrier protein versus this carrier protein, they might have different pickup speeds. So this one picks up sodiums, this one picks up calciums. This one picks up sodiums at a way faster rate than this one picks up calciums. So that means we're going to have more facilitated diffusion through this guy over this guy. So once again, facilitated diffusion, the movement of something from a high concentration gradient to a low concentration gradient using carrier proteins, but does not use energy. Next, we're going with osmosis. Filtration is the last passive transport. It's really quick. Filtration. This is movement based on pressure. So, for example, our kidneys, or you know that your kidneys filter things. So when you have a ton of stuff in your blood is going to filter that out due to the pressure. So if this is a blood vessel, here's my blood vessel, and I've got all these different things in it. When I get to my kidney, the kidney's going to want to filter them out. And because I have all these things in here, I'm going to get more movement. But we've also seen in a chemistry lab, here's I think this is called an Erlenmeyer flask, and you put your big SEP funnel in there, and then you put your paper up top, and you put your solution there, and it leaves a crystallized thing here and liquid down here. So this is gravity filtration, right? Gravity is a pressure, a downward pressure that exists, or a downward force that exists. So this is just another example that he used. So that's filtration. Filtration is movement, diffusion due to pressure. Cool.